All right. Hi guys, Professor Ruggieri here. I'm going to be doing the setup for, now we've already inoculated the initial cultures, but we're going to be setting up for the isolation and spread plate technique lab. So for this, you're going to need your clean snap cap test tube. You're going to need your broth cultures of your Serratia marcescens, and we used this in the last one, the aseptic technique. And then you're going to need your culture of Micrococcus luteus. You're also going to need a sterile nutrient agar plate, your loops, 2 sterile transfer pipettes, okay, and your candle, as long as it'll stay on. So a couple things to do before we actually dig in, but one thing I'm gonna ensure is that we mix together the bacteria before we label our plate, so it has a few seconds to sit. You have this sterile tube. Now, even if you wash it and reuse it, which I don't recommend, but hold on to it for now because I have to check that you don't need it later in the kit. I'm gonna write on it so there's no confusion. M lute, okay, 10 M lute plus 1 S mar, because in the protocol, you're gonna put 10 drops of the cloudy Micrococcus luteus culture in here and 1 drop of the Serratia marcescens. Why? Because M. luteus doesn't grow as well. The S. marcescens has a competitive advantage. This is going to be our mixture tube of bacteria. Okay? So I'm going to get my transfer pipette ready. Okay? Make sure your alcohol disposal is close, but not too close to the flame. And my flame keeps going out, so I'm going to just pretend that it's there. Okay? I'm going to open up my tube, and I know it tells you to hold the lid in your hands. If you want to, as long as you've cleaned your surface, you can put it top side down, right? The side that doesn't touch the tube, just for a few seconds. You're going to close up your M. luteus tube, mix it up so all the bacteria is pretty well mixed, okay? Open this up, flame the mouth of the tube, hold the cap in whatever hand you're holding the tube in, squeeze the bulb, okay, and draw up some M. luteus solution. And then you're gonna put 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 drops or so. Squirt the rest back into the tube, put this in the alcohol solution, flame the mouth, okay, put this back. And I know in the manual they may say don't return the bacteria to the tube. They didn't give you very much, so go ahead and put it back in there and then flame it. Okay, you wanna act quickly because this is open. Then you're gonna take your next sterile transfer pipette, always grab it and open it bulb side out, okay? Make sure that you mix your Serratia marcescens, flame the loop, okay? Draw up just a little bit of this, you only need a drop. Put 1 drop in there, put the rest back in, put this in your alcohol container, flame the mouth, put this back over here. And then you're gonna put the tube on this, okay? Just kind of flick it. There's not a whole bunch of volume in there, you don't need it. Leave it in your rack for a second while you label your plate, okay? You don't necessarily need to, but it helps visually to divide right on the bottom, right? You're always gonna write on the agar side. Let's cut it into thirds, right? So like a little peace symbol. Why are we doing this? Because this lab is isolation and spread plate technique. Okay, so isolation, you're mixing a bacteria that should be pinkish in color with one that should be yellow. Luti means yellow. Okay, so the spread plate part of it is you want to put the densest, you're going to dip it into the broth, the mixed broth right here, and streak. And I'm going to label my my thirds, 1, 2, 3, so I know which one comes first, next, and then last. In the number 1 pie piece, I'm gonna dip a sterile loop into there and then streak like a zigzag all across 1. I'm gonna put that loop in here. I'm not gonna go back into my broth solution. I'm gonna take a sterile loop and drag a bit of the culture from third 1 into third 2. Streak, drop that into the alcohol, grab a sterile loop, then drag from 2 into 3, and then sterilize. Why do I do that? Every time I'm moving from 1/3 to the next, I'm diluting the culture. This is an isolation lab too. So our goal is to get one of these pie pieces where there are distinct and separate yellow and pink colonies. So we're able to isolate one or the other. Okay, and one thing I almost forgot to do, label my plate, right? This is M lute, and I'm going to put 10 to, put the little ratio, the sign, the colon, S mar 1. So it's 10 parts M. luteus to 1 part S. marcescens. Then I'm going to put my initials, the date, 5/31, and then NA for nutrient agar. Okay, so I'm taking out my sterile loop. I'm going to open up my tube of mixed broth, just give it another flick or two. Open it up, go down, get a loop full of the mixture, come back up, put the cap on my tube, you can fix it later. Open it up, make sure you're in third one, and go back and forth, back and forth, back and forth. Okay, put that in here. Get out another one. Go from 1 into, whoops, I went the wrong way. Let me go back this way. 1 into 2. Okay, so I may get a little bit of an error there. Okay, put this in the sterilizing solution. Then I'm going to turn it. And I'm going to go from 2, drag a little bit of the culture. It's hard to see, I know. A little bit of that dampness from 2 into 3. Streak in pie piece 3. Sterilize and close up. Okay? Then you're going to go put this in a safe spot where you also have your aseptic technique plate, and we'll wait to see the results in a day or two.